We explore how war, colonialism, and migrations have reshaped Somali's genetic map. We discover how diverse populations have mixed over time, creating a unique human mosaic and uncovering the lasting impact of conflicts and historical events. This not only made food safer, but unlocked new nutritional value, such as increased bioavailability of calcium and iron from fermented camel milk or dried fish. Interestingly, fermented foods may have shaped not just the gut, but also cultural identity. Traditional fermented milk drinks or dried meat preparations like atka helped preserve food for long migrations. These methods, passed down through generations, influenced microbial transmission patterns within communities. Mothers passed not only food knowledge to children, but beneficial microbes too, both through diet and physical contact. The role of water must also be considered. In arid Djibouti, where fresh water is precious, microbes found in ancient water containers reveal both contamination risks and adaptive responses. Some modern Somalis and Djiboutian populations show increased resistance to waterborne pathogens like Giardia and Cryptosporidium, suggesting an evolutionary legacy of exposure. These adaptations can be traced through both human DNA and the microbial genomes preserved in ancient water sources. By piecing together this microbial mosaic, scientists are not only reconstructing ancient meals, they are uncovering how diet, environment, and microbes collectively sculpted human evolution in Somalia and Djibouti. This is not a passive story of survival, but one of partnership between humans and bacteria, food and genes, culture and biology. In the end, microbial DNA shows that the story of food in Somalia and Djibouti isn't just about what was eaten, it's about how life adapted around what was available. In the fermented milk of a nomad, the grilled fish of a coastal family, or the foraged greens of a desert traveler, we find the code of adaptation, written not only in the genome, but in the microscopic life that moved and thrived with it. When the Europeans formally established their colonial presence in the Horn of Africa, Somalia was divided into three. The French formally established their colonial presence in what is now known as Djibouti in 1884. The British Empire in the north and was known as British Somaliland, and the Italians in the south, which was at the time known as Italian Somaliland. They were entering a landscape already rich in cultural and genetic diversity. But colonialism brought a new wave of genetic influence, this time from Europe and other parts of the French Empire. While the legacy of colonial rule is often measured in language, infrastructure, and political upheaval, it also left more intimate, enduring traces written directly into the DNA of Djibouti's coastal and urban populations. French rule over Djibouti, then known as French Somaliland, created new networks of human movement. European administrators, military officers, and merchants arrived from Europe, North Africa, Madagascar, and West Africa. Many came temporarily, but some settled and formed families, leaving behind a lineage that would blend with local populations. These unions, whether consensual, strategic, or the result of colonial hierarchies, contributed to a small but noticeable infusion of European and Afro-Europeans' ancestry in the gene pool. Genetic evidence collected from Somalia, Djibouti City, and surrounding towns shows elevated frequencies of certain Y chromosome markers common in southern Europe, especially haplogroup R1b. This lineage, widespread in France and parts of the Mediterranean, is rare in the broader Horn of Africa, but appears in select Somali and Djiboutian families with known historical ties to the colonial period. These findings align with oral histories of families descended from European officers, colonial soldiers, or civil administrators. The European colonial system also relied heavily on African troops, many of whom were drawn from Senegal, Mali, and Guinea. These men, known as tireurs sénégalais, were stationed in Djibouti for extended periods and sometimes started families with local women. Mitochondrial DNA studies have found West African maternal haplogroups, particularly L2A and L3E, in parts of urban Djibouti, markers that are rare in the region, but common in West Africa. These results strongly suggest the integration of West African women and their descendants into Djibouti's post-colonial population. Autosomal DNA analysis provides a broader picture. Among certain individuals in the capital and port towns, small segments of DNA show affinities with Western European, North African, and Malagasy populations. These mixed lineages reflect the diversity of people brought into the colony: French settlers, Algerian clerks, Moroccan craftsmen, and Malagasy servants. The colonial port acted as a hub of human convergence, not just commerce. Interestingly, these genetic traces are often invisible within cultural identity. Most descendants of colonial unions in Djibouti identify as Afar, Somali, or Cushitic and maintain traditional languages and customs. But their DNA quietly carries the evidence of this layered past. Family names, lighter eye color, or faintly different facial features may hint at distant ancestry. Yet, the dominant social identity remains deeply rooted in local tradition. The French also introduced European medical practices, including early genetic screenings and blood typing. These initiatives, although often motivated by colonial control or scientific curiosity, produced data that researchers are now revisiting. Blood group studies from the 1930s, for example, documented unusually high rates of Rh-negative blood in certain Djiboutian communities, more common in European populations and now understood as a possible colonial genetic legacy. Further implications are found in disease resistance and health-related traits. Gene variants such as HLA-B27, associated with autoimmune disorders, are more prevalent among mixed-lineage urban Djiboutians than in rural populations. While the exact origins of these variants remain under study, some researchers believe they may reflect European or North African introduction. In addition, certain metabolic genes influenced by European diets and alcohol consumption appear in trace frequencies among modern residents, offering insight into how colonial-era lifestyles influenced genetic selection. Yet the most profound effects of the French presence may lie not in the quantity of DNA they contributed, but in how they altered patterns of gene flow. Colonial policies emphasized urban centralization, wage labor, and formal education, systems that encouraged people from diverse backgrounds to live and work in close proximity. Intermarriage between different African ethnic groups increased in cities, sometimes encouraged by the need for social advancement under French rule. This resulted in a secondary wave of admixture, not between locals and foreigners, but between local groups themselves. For example, mixed Afar and Issa lineages became more common in urban environments where colonial administration diluted traditional rivalries and boundaries. This internal admixture is now visible in autosomal DNA, where formerly distinct populations show increasing genetic overlap, another layer in Djibouti's complex human mosaic. Modern Djibouti continues to reflect this hybrid history. French remains one of the official languages, and dual citizenship with France is not uncommon. Educational and military exchanges maintain the connection, and families with Franco-Djiboutian heritage contribute to a sense of cosmopolitan identity, especially in the capital. Yet, despite this legacy, Djiboutian identity remains strongly anchored in Somalia, African and Islamic traditions. In many ways, the French connection in Djiboutian DNA represents both disruption and continuity. It brought new genes into the population, but it also reshaped existing dynamics of marriage, migration, and kinship. Today, as scientists map the genome of the Horn of Africa, these colonial layers remind us that identity is neither pure nor fixed. It is shaped by movement, power, and history. The story of colonial DNA in Djibouti is not just about foreign bloodlines. It's about how people absorbed and reshaped them, about how children of empire grew into citizens of a new republic, carrying within them the complicated heritage of both colonizer and colonized. In every gene that bears the mark of a foreign name, There is also the imprint of local survival, adaptation, and enduring pride. Conflict has always been a force that reshapes societies, but its influence extends beyond borders and politics. War uproots families, redraws maps, and reshuffles populations. In Djibouti, a country that has witnessed regional warfare, internal skirmishes, and refugee influxes, these upheavals have quietly altered the genetic landscape. The result is a complex mosaic of genetic shifts, driven not by trade or tradition, but by displacement, crisis, and survival. Though Djibouti itself has seen relatively few prolonged internal conflicts, its location near conflict zones has made it a haven and a crossroads for fleeing populations. Its proximity to Ethiopia, Somalia, and Eritrea, each with their own histories of war, has made Djibouti both a place of refuge and a corridor for human movement. These demographic surges have brought new genes, new languages, and new identities into the region. One of the earliest and most significant episodes of genetically relevant displacement came during the Ogaden War of 1977-1978. This conflict between Ethiopia and Somalia over the ethnically Somali Ogaden region sent waves of Somali-speaking refugees into Djibouti. Many of these displaced individuals were absorbed into existing Issa communities or settled in newly created neighborhoods. Genetic studies today show a slight but measurable increase in Somali DNA markers in post-1970s Djiboutian generations, especially in urban areas like Balbala and Olemsabila. More broadly, Y chromosome haplogroups such as EM35 and TM70, common among Somali populations, saw a noticeable rise in frequency during the decades following the conflict. These paternal markers, already present in Djibouti due to shared Cushitic ancestry, became more pronounced in urban centers as refugee communities integrated through marriage and migration. While these changes are not dramatic, they provide a genetic echo of geopolitical disruption. Eritrean conflicts during the 1980s and 1990s also had demographic consequences for Djibouti. During the long war for Eritrea's independence from Ethiopia, thousands of Eritreans, many of them Tigrinya-speaking Christians and Muslims, fled southward. Djibouti, with its established port and colonial legacy of relative openness, became a temporary home for many. While some refugees eventually moved on or returned home, a significant number remained and assimilated, contributing to the genetic diversity of coastal Djiboutian populations. Autosomal DNA comparisons of urban Djiboutians reveal increased genetic affinity with highland Ethiopian groups, particularly Tigrayans and Amharas, in contrast to earlier generations. This suggests recent admixture due to refugee influx and intermarriage. Even more interesting is the maternal side of this genetic equation.